Dear participant, I welcome you in the 21st day new session of the countrywide classroom teaching program CWCP 2021. And today we are continuing the previous lecture of 20th day delivered by Dr. Ashok Bankar, and topic is viral infection. Now I request Dr. Ashok Bankar sir to continue his session from the previous session 20 day of uh, the CWCP. Ashok Bankar sir, the
Yes,
session
sir.
is over to you. Yes, sir.
Okay. So thank you for your opportunity and invitation. I again thanks all the participants for your patience. In previous lecture, as I was explaining about the AIDS and as well as the hepatitis A, now the next session is then we study different symptoms and how to tackle with the HIV, how to prevent. In medical science, it is always said that the prevention is better than cure. So that is true about the AIDS as well as the other diseases also in hepatitis also, hepatitis A and all that. So although there are some treatment for the AIDS, but it can only increase the life, but we cannot 100% cure the HIV. Then we studied about the virus, viral hepatitis, that is hepatitis A. Then we understood the structure and basic things about the hepatitis A viruses and how it transmitted, their incubation period, their clinical symptoms we studied in previous lecture. Then what kind of how we can diagnose it and prevention measures we studied in previous lecture, and the control of transmission also we studied. Vaccines, what kind of the vaccines available for the hepatitis A, that we studied. And in this way, we found that these live attenuated vaccines and other vaccines are very valuable, very important for to control the hepatitis A. So, like hepatitis A, another one of the most important virus or the disease, and that is the hepatitis B. So, hepatitis B is mostly caused because of the hepatitis B virus. Okay, so I already told you that what you mean by the hepatitis uh, B. I already told you that what you mean by the virus. Okay, as I told you that the virus is nothing but the uh, it is a particle. And then we studied that what this particle contains simply capsid proteins as well as the genetic material, either it is RNA or it is a DNA. Okay, so this hepatitis B virus is an envelope virus. So what do you mean by the envelope? So they have envelope, outer covering, then capsid. Okay, so there is a genetic material, then capsid is there, and then again there is a one more envelope is there. Okay, so this is an envelope virus. Okay, so this envelope virus contain, that means genetic material is very well protected by this envelope, and it contains the double-stranded circular DNA genome. Okay, so thus this virus, hepatitis B virus, is classified under the family, that is the hepatinovirus family. The Now, what is the main role of this virus? This virus interferes with the functions of the liver. That means it is a clearly indication that this virus is very specific for the hepatocytes. Okay, so hepatocytes, that is the cells of the liver, and they will that means this virus they are very specific for the hepatocytes. Okay, hepatocyte cells, and this indicate that surely this virus is going to be affect our liver. Okay, so it will interfere the structure of the liver, it will interfere with the function of the liver. Am I right? So virus here interfering with the whatever the functions of the liver, and thus the immune system is then activated once this virus will take entry in the body, and they will produce some specific antibodies against this virus. So our immune system will start to combat or fight with this virus, and then later on it will eradicate the all the infectious agents like virus and associated or co-infections. Our immune system will try. Okay, but if our immune system is little bit weak or fail, so what will they happen? The next thing is that there will be the pathological damage. So liver become inflamed. Implant means it will be swollen and the structure will be little different, and liver will be little damaged. Okay, so like that the liver how the interference with the viruses. So like that, all of you know that liver is one of the very important organ in our body, and all the detoxification process which is happening in the liver. So liver is very crucial and very important for the life, and that is the reason we have to take care about our liver, and we need to understand this virus very well. So this hepatitis B virus may cause up to 80% all cases, that is a hepatocellular carcinoma worldwide. Okay, so hepatocellular carcinoma. So all of you know that the virus have ability to develop and interfere with our genetic material, and it may be result into the liver cancer also. Okay, so we have to be very smart, and we have to be very I mean smart enough to control this virus, and we should also have the some knowledge about this virus, so you can help to your family members as well as the society members. So as a microbiologist, it's your role, as a biology student, it's your role to make aware to our family members as well as our society. It's a role of each and every scientific person. Okay, so first we'll understand the structure of hepatitis B virus. So how this hepatitis B virus look like, and what is some special characteristics of this virus. So hepatitis virus is, as I said, it is a double-stranded DNA virus, and the genome is very much compact and very well organized, and the genome is a circular double-stranded DNA, and the replication occurs by the reverse transcriptase enzyme. So here reverse transcriptase enzyme is very much important for the replication of the viruses. Okay, so why reverse transcriptase, I will explain it later on, but this enzyme is very much important in this virus. So this virus is very small and circular, and almost it contained 3,200 base pair size, that means genomic size, and its DNA size. They can code for the products. You know that all the information is on the DNA, because DNA is a genetic material. So what do you mean by the genetic material? Because it code for all the characteristics. So now here viral characteristics is the viral product, all the product. What is that product? Either it is DNA, or it is a capsid, or it is a other proteins. These all this complex multi-particle structure is all this coded by the DNA. So here DNA is very much important. So hepatitis B virus is a if you see the size of the hepatitis B virus, it is a 42 nanometer. So what do you mean by the 42 nanometer? So nanometer is very small. 1 micrometer is equal to 1,000 nanometer. So you can imagine that how small it is virus. Okay, so 1,000 nanometer. 1 micron means 1,000 nanometer. The size of the bacteria, if you see the 2 micron, depending upon the what kind of the bacteria it is, 1, 2, 3, like that, and some bacteria are even longer than that. So you can imagine that the nanometer size is very small. So if you want to see this if you want to observe this virus, you cannot use your normal microscope or light microscope or any other microscope. But for this to observe this virus, you need very high resolution power of the microscope, and that microscope is nothing but it is a electronic microscope, that is called as a TEM, transmission electron microscope, because resolution is very much high. Okay, 1 lakh and 2 lakh. So it's become visible, a virus become visible under the transmission electron microscope. And not only you have to take the virus and you see under the microscope, there are several some processes. Okay, you have to give some processes to the virus, then and then it become visible. Okay, so it is not like that you are taking sample and directly observing under the electron microscope. No students. Okay, so lots of process techniques are required if you read some research papers. So students also try to habit to develop that you should not only dependent on your textbook or your reference book, but you should also learn to refer the research paper, what is the recent information is coming in the our field. that you should read properly, and that will be very useful to you in your future to qualify SECNET exams, because you should know how you should know the different techniques and the basic things. You should know that. Okay, so you have to study the basic things. So what is the basic things? For example, here I am saying that the size of virus particle is a 42 nanometer. That means you should understand what you mean by the nanometer. That is a basic thing. For example, if I'm saying the nucleocapsid core, it is a 27 nanometer. Then what is nucleocapsid? Okay, so nucleus is a, I mean, enclosed in that capsid, that is called as a nucleocapsid. So it is a protective capsid protein. Okay, it is a protective capsid for the DNA, and that is called as a core core antigen, and which is surrounded by the lipoprotein coat. So what do you mean by lipoprotein coat? So like that you have to understand lipoprotein, that means lipid is there, protein is there. It's a combination of lipid and the protein. So that coat is called as an envelope, and that's why we are saying that this is an envelope virus. So all these basic things, student, you should understand. What do you mean by lipid? Then you have to understand the fatty acid. Okay, you have to see the structure of the fatty acid, how one fatty acid linked to another fatty acid, and how the lipid is formed, and why there is a different structures of the lipid, how protein is formed. So protein basic unit is amino acid, how one amino acid attached to the another amino acid, and how protein structure is formed. So that you need to understand. If your that basic is okay, then you can easily understand lipoprotein code. Otherwise, what will happen? You'll just memorize it, and you'll write your paper, you'll get the good marks. But you, my dear students, I'm telling you that memorization and writing the paper is not the knowledge. Okay, tomorrow you may forget. But if you understood the story, but if you understood the basic, if you understood the basic biochemistry of the protein and the lipid, you'll never forget in your life. And that is important. So acquiring knowledge is important, not only the reading, but understanding is very much important. So novel thinking is a necessary, and then you can put your idea and you can do the very good research. So student, it's my role to explain that how you can prepare yourself for the future fight or for future comeback. You know that the coronavirus is not only the challenge, but in future many viruses will become. So for that you have to be ready, and that's why you should have the good knowledge of your subject. Okay, so viron here referred as some this viron, sometimes it is also called as a Dan particle. So why it is called as a Dan particle, DANA? Because it is a double-stranded DNA. That's why sometimes people refer it as a Dan particle. So in future, suppose the word comes, the Dan particle, you should understand yes, it is a hepatitis B virus. Okay. So core antigen is located in the center. That means in the nucleocapsid. And core anti, and so they are called as a antigen. Core antigen because it is a, and then it is called another antigen, that is a e antigen, that is a secretory antigen. Okay, so what do you mean by the antigen, students? So antigen means it is nothing but any material which is foreign to our body. They are called as a antigen, which is a complex and high in molecular weight. Okay, for example, any bacteria entering in our body, that is antigen. Okay, their protein is antigen. There's some specific part, maybe the antigen to our body, and then our body will produce the antibodies against these agents. For example, it may be the pollen grain or anything you inject in the body that is not part of our body. Okay, so that will be considered as an antigen, as a foreign material. Okay, so foreign material is a antigen, and vaccine is prepared by the same from the virus. You have to inactivate that virus, or you have to remove the genetic material, or you have to only take the protein part of that virus, and then you have to inject, because it is not infective. So when virus is active or functional, only when there is a proper structure is well assembled and proper environment is there. But if the structure is a change or you disorganize, if the structure is disorganized, then virus cannot be active or virus cannot be functional. So here you can see the structure of this virus, hepatitis B. So here at the center, here you can see that the red color, which is shown that the DNA double strand. Okay, so double stranded DNA is there, and just wait. So here you can see that the so here you can see that this is a genetic material DNA, double-stranded, and some part is you can see that it is a single-stranded. Okay, so that's why it is a partial single-stranded. Okay, so DNA is a partial single-stranded, but if you see the other region, the DNA is a double-stranded. Okay, but only this part is a single-stranded. Then you can see that this is a capsid. This is called as a nucleocapsid. Okay, around whatever the proteins which is around, and this is called as a nucleocapsid. So as I said, the core antigen. So core antigen, these are the core antigen. This is called as a core antigen because this antigen, these proteins are acting as an antigen, as a foreign material for our body, and against this antibodies can be produced. So core antigen because they are in the nucleocapsid. Okay, that's why it is called as a core antigen. Concept is clear. Hepatitis B core antigen, HBCAG. Okay, this is called as a HBCAG. Okay, the next is you can see this is a dot 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 something secretion like structure. So these are nothing but the secretory proteins, and these are called as a secretory antigen. Okay, so that's why they are called as a E antigen. Okay, hepatitis B secreted E antigens. So now you understood what you mean by the core antigen. This is a core antigen, okay, protein, and this is a secretory antigen, that is a E antigen, and this is a genomic material, that is a DNA double-stranded, but at this with partial strand also, single strand. Okay, so now you understood this. So this is very basic structure. And after this nucleocapsid, now envelope is there. Okay, so outermost envelope is nothing but it is the outermost covering of the virus. Okay, so this outermost covering, and this part is called as total this part is called as a nucleocapsid, viral capsid, and viral capsid is now covered with this envelope, and that's why this virus is called as a envelope viruses. Now what is there in this envelope? So here you can see that, can you see this is a membrane like structure? You understood the in membrane what is there, lipids are there. Okay, so here you can see that the lipid like structure, here you can see the round, and these are the, this is head and this is a tail of fatty acids. So head is called as a hydrophilic and the tail is called as a hydrophobic, and that's why organization is like this. So head is towards, so heads are arranged like this. So this is a membrane like structure. So this is a membrane of these viruses. Now this membrane is derived from the host. When this virus enter into the host, at that time, whatever the covering, this covering is from the host. So this is a part of the host. Now what is there in this envelope? There is a lipid. So here you can say this is a lipid, and then what is this mushroom-like structure? This is nothing but this is again the antigen, and they are called as a hepatitis B surface antigen because it is a at the surface, and this antigen plays very important role for the attachment to the host, to recognize the host, and that's why the specificity is there. It's like lock and key model. Okay, when you are using the key and then you are opening the lock, so like that in our body there are receptor, and they are called like the lock, they are locked like this, we can call them receptor. And once the virus interact with the lock or interact with the receptor, automatically it open the lock because it fit in that area. So like that this virus, whatever these are the structures, they recognize the receptor. Where is the receptor? Now this receptor will be on the hepatocyte cell, that means in the liver. Okay, so target of this virus will not be kidney, okay, not be heart, but what will be the target? Target will be the liver. Why? Because of this specific structure. So here students, structure plays very important role. If you destroy the structure of the virus, virus will be inactive. Okay, if you destroy the structure of eye, you cannot see. Okay, so that means structure is important. If you change the structure of a heart will non-functional. So like that, structure is very important, organization is important. So proper organization of this molecule is very important. Proper organization of protein, proper organization of the DNA, proper organization, all these materials are important for the to remain active to this virus, to functional. in the body of human being. All these organization, proper organization structure is important of the virus. If you destroy this structure with the help of some chemicals, then this virus will be inactive. Then you can see that this virus is inactive and active. Okay, when it is active, when a structure is properly well preserved. When it is inactive, like coronavirus, if you use alcohol or a detergent, it structure will be destroyed. Then this lipid membrane will be destroyed with the help of the detergents and alcohol, and that is the reason, students, virus is inactive. Okay, so I hope you understood the spherical structure of the virus, hepatitis B. And sometimes, students, this virus is also elongated. Okay, it is spherical, but sometimes it may be elongated. It is depending upon the requirement of the viruses, or the some genetic, there must be some changes in the viruses according to the need. If you take the section of this virus, how it will be look like? If you take the section, as I said, it is a 42 nanometer in size. Here you can see measure the length. How this measure length is measured? This length is measured with the help of the TEM, transmission electron microscope. In that you can measure the there are lots of softwares available, and the softwares are installed. So live you can measure the length of the virus, length of the bacteria, length of the fungi, length of the spores. Okay, so all this is a miracle of the microscopy. Here you can see that the core antigen, I already told you that the core antigen, and there is one of the special enzyme is also there, that is called as a protease kinase, and these are the again the surface protein, and they are called as a surface antigen. So specifically, three type of antigens are recognized, or three type of the more important proteins has been recognized or identified in case of the hepatitis B virus. Which are these antigen, friends? So antigen number one, that is a surface protein. So this is a surface protein. Okay, here you can see this is a surface protein, and they are called as a HBS antigen. S for surface. Okay, so in future you remember it. S means surface protein. Then inner core protein. So here you can see that the inner coat. Okay, so sorry, this is a DNA, and this is a inner coat protein. So these are called as a core antigen. Okay, now clear. That is called as HBCAG. C for core. So inner core protein, or you can say that the core antigen. Third one is the secreted protein. I already told that the small dots are present inside. Here you cannot see that such a small dot. I already shown in the previous figure. Here you can see that these the small dot, these are called as a secretory proteins. So they are secreted. That's why they are called as a E antigen. HB for hepatitis B. Hepatitis B E antigens. Okay, so now you understood the basic structure of this hepatitis virus B, and let's see how this virus replicate. So replication strategy. Okay, so first thing is that this virus. Okay, so this HBV viron. Okay, this particle. That's why they are called as a viron. They binds to a receptor. Your receptor plays very important role. Now, friends, where this receptor is present? So this receptor is present on the surface of the hepatocyte. Okay, whatever the hepatocyte or whatever the liver cell is there, on that hepatocyte, there will be the some receptor. Okay, we can call this like the lock of our home. Okay, so if you remove or if you want to open the your house, what will happen? You'll take the key. Okay, and then you'll fit this key in the lock, and then automatically lock will be open. And once the lock is open, house is open for the entry of the any person, those have that key. Okay, so house doesn't discriminate between the different persons. Either it may be the owner or it may be the thief. Okay, so house don't know or lock don't know this is a thief and this is a owner. So only principle is that if the key is matched, lock will open. Like that, this virus will goes and bind to the receptor, the surface hepatocyte cells. And so what will happen, students? Automatically lock will open. Which lock? For the entry of viruses. because our body or our cell doesn't know this is a dangerous person or this is a dangerous viron. Okay, so they doesn't recognize, surface receptor doesn't recognize, and then after binding automatically lock is open and then viral nucleocapsids enter the cell. Okay, so virus got the entry and reach the nucleus where the viral genome is delivered, and then it goes and goes to the target, and automatically the all the protein and all this will be dissolved, and the naked genetic material or DNA will be delivered to the nucleus. Okay, so in this way we can in this way the virus take entry. After the entry, then what will happen? So then genetic material is double-stranded DNA. Then it will be transcribed. So transcription will be takes place. So what will happen? Number of mRNAs will be formed. Okay, mRNA copies will be formed, and these mRNA copies. Okay, what will happen? DNA to mRNA. It is called as a transcription process. So all of you know that the three central dogma in the cell. Okay, in eukaryotic cell or in prokaryotic cell, what is this central dogma? That is a DNA replication, RNA formation, that is a transcription, and then from transcription, another is a protein synthesis, that is called as a translation, students. Okay, so you know the DNA replication, transcription, and translation. All these three things are important. Now, what will happen? Now, DNA is there. After the DNA replication, what will happen? mRNA need to be formed. Okay, so once the mRNA will be formed, then translation will be happen. After the translation happens, so protein coat and proteins, everything will be formed. Now everything is formed, proteins are formed. Now cell want this virus want the genetic material, and their genetic material is a DNA. Okay, so how DNA will be again formed? So they form a mRNA, and from mRNA now they have secreted the protein, that is a reverse transcriptase enzyme. So this reverse transcriptase enzyme will go and bind to the mRNA and will convert them then to the DNA. That's why it is called as a reverse transcriptase. What do you mean by the reverse? DNA to RNA is transcription, but RNA to DNA is a reverse process. Understood, students? DNA to RNA is called as a transcription, but RNA to DNA, it is called as a reverse transcription, and that can be can be done with the enzyme, and the name of that enzyme is called as a reverse transcriptase enzyme, because it convert RNA to DNA. So like that, there will be number of RNA molecules will be synthesized and formed, and now they are converted into the DNA. Now viron having for the assembly of viron having the lots of protein and lots of DNA. So there will be the assembly of the viron, and then viron will be matured and it will be come out, and some DNA will be integrated into the host genome and causing as a carrier. Okay, they will remain as a carrier. So if you donate the blood, or at that time this carrier can carry the gene. So diagrammatically we'll see the how this virus interact with the liver cell. Suppose for example, student, this is a liver cell. Okay, so this is our smart liver cell, and this liver cell having the at center there is a nucleus, and then in the nucleus inside there is their genetic material. Understood? This is a basic structure of the our liver cells. You know that in any cells, protein plays very important role. Our bodies also require good amount of the protein. Okay, and that's why the protein is important. So this is a liver cell. And now what happened? Virus doesn't know. As I said, it is a particle like dust like particle. Okay, virus doesn't know that I have to come and go to the liver. Okay, but what happened? By accidentally we are catching these viruses, because of our activity, because of some environmental factors, then virus accidentally come in contact with this liver cell. Okay, virus doesn't know where to go and how to survive. Okay, but it is like the bombarding, simple attachment of the virus. Virus doesn't know, virus don't have the brain and that knowledge that I have to go to the liver cell. But because of the structural specificity, the virus now, if virus comes in contact with the liver cell, what will happen? This virus will recognize the receptor, and these receptors are on the surface of this liver cell. And because of this receptor, the structure is fit. Okay, here you can see the structure is fit, and then it will open the door of the liver cell. Okay, and once it open the door of the liver cell will be activated, some conformational changes will be there, and they will take this virus inside. They will start the process to take the virus inside, and in this way the virus will take entry inside the cell. Now the next job of this virus is that now to remove all this capsid and all the protein and everything, and then he want to send this naked DNA genetic material into the nucleus of the cell. That will be the next action of this virus. So in this way what will happen, then it will reach to the nucleus, and then it will be having associated some proteins and DNA polymerase enzymes and other undetected proteins and enzymes. They are secreted, and in this way now they will release their genetic material, and then the process will be takes place. So what will happen, transcription will be there and translation will be there. So after the transcription, mRNA is formed, and from mRNA translation will be takes place, so protein will be formed. Okay, and in this way core protein will be formed. So all these process are transcription and translation will be happen in the nucleus with the help of the machineries which is present in the hepatocyte cell. Okay, so they are utilizing all the facilities, and that's why it is called as a obligate intracellular parasite. So why virus is called as a obligate? Obligate means strict. Intracellular, that means inside the cell. Obligate intracellular parasite. And why it is called as a parasite? Because it is using all the facility of the host, and it is using that facility multiplying and again damaging the host, damaging this cell. And that's why the parasite get benefited, because they multiply and host is harm. And that's why hepatocytes will get harm and damage. And that's why this is called as a host, and this is called as a parasite. Now you understood host and parasite relationship between the any thing in my I mean virus and human being. So virus is a parasite, human body is a host. Bacteria is a parasite, human body is a host. Fungi is a parasite. If it is infective, then human body will be host. If the plant is a host, plant viruses will be infective. Okay, they will infect. So plant viruses will be the parasite and plant will be the host. Like similarly, like in animal also, some viruses are specific for the animal. So um, uh, animal will be host and the virus will be the parasite. So in this way what happen, once this process is a form, another important thing is that the proper assembly. So genetic material DNA is a form, these proteins are formed, core proteins are formed, and then there is assembly, and after proper assembly there is a formation of the particles. So there is extra surface proteins are also produced, and because of that ground glass like structure will be appear in the blood when you see under the microscope. So such kinds of the structure you can identify for the diagnosis purpose, and therefore when people are repeating the experiment, and with the help of their experience working in the laboratory, they can easily identify the infection of the hepatitis B under the microscope. And that's why we need a experience, we need a expertise, we have to carry out the experiments also. That is a reason, because we should know how to handle the microscope, we should develop all the skill. Why I am telling all these things, students, all of you know that who is the father of microbiology? So Anthony von Leeuwenhoek. Why that person is called as a father of microbiology? Even he was a merchant, he was a merchant of glasses, and because of his skill, because whatever the microscope he has prepared, that microscope, under that microscope only the Anthony von Leeuwenhoek could see the microorganism. If you give this microscope to the other person was not able to see the microorganism under the microscope. The reason is that because he was very skillful, and therefore students, obtaining mark is not the skill. Okay, it's just your degree. Okay, getting mark, it is just you are saying that I am first class, but what about your skill? You are saying I am in distinction, but what about your skill? Have you developed your skill? So if you don't develop your skill, you have to develop your skill. You have to concentrate on your skill. And all these skills are important. Your way of presentation, your way of behave, everything is very important when you are learning the science. Your thinking level, your novel ideas, all this is important. So question yourself. Whenever you are studying, question yourself, and then you will understand the concept. If you are only reading and memorizing it, that is not the science. So meaning of science. So what is the meaning of science? So science words itself is not an English word, Hindi, Marathi, or any Tamil or Gujarati. No. So science word is from the Latin language. The meaning of science is to know. I don't know, I want to know. That is a science. Okay. If you don't know, student, that is not your problem. Then what you have to do? You want to know. Okay. So otherwise you will feel that it is miracle. If you don't understand the mechanism, when first flight, okay, first aeroplane was fly, people thought that it's miracle. But today, do you think that it is miracle? No, because you understood. Flying aeroplane is not miracle because it is based on some principles. So similarly, when you understood the concept, you feel that yes, you understood, then you don't say it's miracle. Why? Because you understood the concept. But if you don't know you don't have that knowledge, you will be surprised. You will be feel that yes, it's miracle. Okay, so this is simple life cycle of the hepatitis B virus. So how it come in into contact with the hepatocytes, and then it take entry, then it releases genetic material into the nucleus, and then transcription happen, then translation happens. Okay, in nucleus transcription happen, after that it is transported outside, and then translation is happening in the cytoplasm, and then reverse transcriptase will come after the translation. Reverse transcription takes place, and then this RNA molecule will be converted into the DNA. Okay, so like that the DNA negative strand and DNA positive strand will link together, and like that assembly will be formed, and then virus will be released outside. So in this way, so now students, here you can see that this is nothing but the membrane. This membrane is from the host, and this membrane is now envelope, working as an envelope, protective in nature, and then it is a release. So once it is a release, this part is from the host, and this is a nucleoside inside. Okay, so like that here you can see that the DNA double stranded, but it is a partial at some space, and then nucleocapsid is there, very well organized. Here you can see that nucleocapsid is formed, and then this is genetic material, and then to this is a total viron is ready to release. This is called as a matured hepatitis B virus. Okay, so this is a matured hepatitis B virus. Once it is matured, it will ready to infect to the other host or other cells. Okay, that's why it is a dangerous. Now, what is the mode of transmission of this virus? So first mode of transmission we'll see, that is a sexual. Okay, so sex workers, as I said, prostitute or homosexuals are particularly at high risk. Okay, they can transmit it from male to female or female to male. Second one is a parenteral, that is most of the health worker, they are handling the blood samples and plasma and RBC and WBC. So at that time they are on the high risk. Okay, so in this way, uh, then third one is a perinatal, sorry, perinatal. That means from mother to the positive mother to the their offspring. Okay, it can easily transfer to their offspring. So this is one of the main mode of transmission in the population. So here you can see that almost 90% of the infected infants become chronically infected because of the mother. Okay, so mother and the infant. So this is a strong correlation for this perinatal transmission, viral transmission. Another one is how it is a transfer to the one host to the another 6%. So that is a child to child, contaminated needles, sexual contact, healthcare worker, as well as the blood transfusion. And that is the reason, student, it is very important when you are donating any blood. Okay, So you should not have the hepatitis B infection, hepatitis A infection, or HIV infection. So that is very important. Otherwise, the life of the another person will be also on the you will there will be the threat to the life of other persons also. So here you can see the distribution. Uh, so, you can see the different percentage injecting drugs, contaminated needles that can be also transmitted, transmit this virus. Then homosexual relationship also 9% they can transfer, unknown reason 31, and another that is a 41, that is a actual reason, that is a perinatal or parental. So who is on the high risk? Okay, so the people from the endemic regions, okay, the babies of mother with, yes, I'm audible.
Yes, sir. Yes, please go ahead.
Okay, so
Yes,
the
sir, you're audible, sir.
so the people from the endemic regions, and in that particular region, if disease is occurring, then they are on high risk. The babies of the infected mothers, then intravenous drug abusers. As I told you in last lecture also, these abusers are nothing but they are called as, you can say them as a self abuser. So what do you mean by the abuser? That means you are cheating to yourself. Okay, so how you are cheating? For example, if I'm sleeping for uh, if I am sleeping for 10 hours, 12 hours, 30 hours, so I'll be lazy. Okay, so this is, I'm abusing myself, I'm abusing my body, I'm abusing my habits. Okay, so these are called as a self abuse. So intravenous drug abusers are nothing but, this is a group. This group they form for the to take the drug, and they are using the common needle. Okay, to inject the drugs. So drug, once they inject the drug, they will get the pleasure, and body again demand every time. So they people become these people are of the habit of that pleasure, and day by day the dose of the drug will be enhanced, increase, because their receptors are demanding more and more drugs. Similar thing is like alcohol also. Okay, if you take the small amount of alcohol, then your body will demand the more amount of alcohol. Then you'll take the more alcohol, then again it will demand. Then time will come, you will drink every day. Okay, because of the demand of the body receptors. And in this way, then the person will only think about their own pleasure. The person will become selfish because their body is demanding like this. And we are saying that that person is bad. Why that person become bad? Because of the their body is demanding, and that person abuse their own body. That is a reason. And this is a reason even you counsel these people, they will not listen to you, because they want drug and drug, because they have that much habit in the body. They cannot survive, or they don't want to be survive without drug. That is the reason. So people with multiple sex partners, so there are many people, they are changing their sex partners. Okay, they are living in live-in relationship, as well as they visit to the sex workers, and because of that they don't have the good knowledge of science and good knowledge about the different infections. So people may get infected with the HIV, hepatitis A, hepatitis B also. So, hemodialysis patient, you know that some patients there, if their kidney is not working, they have to go for the dialysis, because you know that students, you are very lucky that you have the good kidneys. The main function of the kidney is to filter our blood and remove the unwanted toxic material from our body, that is a nitrogenous waste material, and thus through the urine, all of unwanted materials will be removed. And thus in this way our kidneys plays very crucial role. So we have two kidneys, and these kidneys are helping, but because of your unwanted habits, your unwanted drugs consumptions, painkiller consumption, unwanted threat is given to the kidney. You are drinking less amount of water, so what will happen? There will be the pressure on the kidney, extra work to the kidney. So whatever the activity you are doing, that will be the extra. For example, if you're taking the lots of drug without reason, okay, some pain is there, you are taking painkiller. So what will happen? You are thinking that now I don't have pain, but it you are giving stress to your kidney. Understood? You are not drinking water, you are giving stress to the kidney, you are increase the problem to the kidney, and you because of that you are drinking a lots of alcohol, so it is again threat to the kidney. You are drinking lots, you are smoking, or many things activity you are doing, so all these things are going to happen to a problem to your kidney, and kidney will collapse, and then you will need the dialysis. Okay, so dialysis is an artificial process, and then your confidence will be loose, and psychological and mental problem will be there, and you have to pay so much money for this dialysis, because your life is on the dialysis. So now you understood the importance of your kidney, students, and why your kidneys are important, why your heart is important, why your brain is important. So it's our job to take care of all these important vital organs, because you cannot replace it by money only. Okay, so your brain, your heart, your lungs, your kidneys, these are the powerful organs, and it is your job to take care of them. It's your job to give them good nutrient. It's your job to exercise them properly, and then they will work for you powerfully, and automatically your immune system will be work nicely. Okay, so healthcare personals which comes in contact with the blood, they will also impact, because they are handling all the blood samples, and by mistakes if there is a contact happening, and that will be create the problem. So resident and the staff members of the institution for the mentally retarded, so these staff members and mentally retarded persons, at that time, because of lack of knowledge and all these things, this may happen. So one question is from the student is that, so how to keep kidney healthy? Very nice question, students. It's very good that you are aware about your kidney. So to keep a kidney, first thing is that you have to drink a lots of water, and you have to avoid the unwanted drugs. You have to control your diet. Okay, what do you mean by the control your diet? That means unwanted. You know that our diet is very much rich in carbohydrate and very less in the protein. Check your diet, students, what you are eating. Understand the diet. So your diet should be rich in protein, and lots of water should be there. Lots of means water should be also in the balance. More amount of water is also harmful to the body. So if you want to keep your kidney clean, so then avoid all the smoking, avoid alcohol consumptions, avoid drug consumptions. Okay, avoid the unwanted antibiotics. Okay, without reason people are taking lots of dose of antibiotic, that is not good for our body. Okay, so you have to develop the natural immunity. Okay, go for walking, do some exercise, so it will, your mental health will be good. If you do the good exercise, walking, jogging, running, anything, cycling, swimming, do anything, that will be help to your all the organ, not only kidney, because all these organs, students, Nikhil, these all organs are interrelated to each other. How they are interconnected? Because circulatory system is blood. Whatever happening in the blood, it will affect on the kidney. Whatever happen in the blood, it will affect on your heart. Okay, because heart is also playing important role in the purification of blood, as well as supply of the blood. So heart, kidney, brain, all these things are interlinked with each other. So you don't have to think only about the kidney, but you have to think about your lungs, you have to think about your heart, as well as the other organs, your digestive system, what you are eating. Okay, if you are eating Chinese, think twice. Why, what I am eating, is it nutritional? If it is not nutritional, 100% work will be given to the kidney, because unwanted things has to remove, filter it. Again, there will be workload to the kidney, because you are not taking the healthy diet. Understood? Understood, Nikhil? So if you want to keep your kidney healthy, you have to avoid the unwanted food material, unwanted drugs, unwanted antibiotic, misuse of the antibiotics, misuse of the painkiller, misuse of the I mean, you don't have to consume alcohol, as well as the smoking. All your habits should be healthy, and proper sleep, that is again important to function all organs properly. And mentally you should be strong. So how you should be mentally strong? just by doing some exercise, meditation, and all these things are going to affect. So I hope you understood. Thank you. So what will happen once this disease will be progressed? First stage, in the first stage, the healthy adults is approximately 2 to 4 weeks. Virus will be in, enter, and incubation period will be 2 to 4 weeks. Again, it is depending upon the person to person. It will be little bit vary. So it is also cause infection to the newborns one. So then active viral replication will be continue later, or there will be no elevation in aminotransferase enzyme level. So no symptoms at the first stage people can observe. Then second stage, what will happen? And inflammatory reactions. You know that what you mean by the inflammatory reactions. In our body, when virus goes, our immune system get activated, and there are some cytokines are released. Okay, signal molecules, interferons are released, and this interferon is giving message to the cells, and then there is a fight against the viruses. So sometimes if your immune system react very high, and that is called as a cytokine storm. Then how to identify you in my body there is a cytokine storm, how the inflammation reaction is going on. So there will be fever in your body, shivering will be there. Okay, so cold will be there, shivering will be there to the body. Then you'll understand that yes, there is a cytokine storm. And then what will happen? Be in this stage there is a cytopathic effect. That means virus will try to multiply and will try to break the hepatocyte cells. Okay, try to break the hepatocyte cells. Okay, and once these hepatocyte cells will be break, what will happen? So again the virus will go and reach to the another healthy hepatocyte cells. Okay, so in this way it will start the developing symptomatic period, 3 to 4 weeks. Okay, so some patients with chronic infection 10 years or more may be required to develop the cirrhosis. Okay, so cirrhosis will be developed. Okay, in the cirrhosis, that is a cirrhosis of liver, it is a not good sign and symptoms, and thus it will be reach into the third stage. And in the third stage, hosts can target the infected hepatocytes, and the viral replication no longer occurs, and simply the HBV DNA levels are lower and undetected. Aminotransferase enzyme level are within the reference range. And in this stage, integration of viral genome into the host hepatocyte genome will take place. Okay, so what happen in the third stage? They will keep some their genetic material into the host. Okay, and still this antigen will be yes, antigen will be there, you can detect. In the first stage the virus cannot be detected. Now the virus will not be there at the first stage, and antibodies to the various viral antigen like the surface antigen, core antigen, and the e antigen will be produced, will be there. And the different factors have been postulated to influence the evolution of these stages. This is depending upon again age, sex, immunosuppression, and co-infection with the other viruses. Okay, so these factors or influence of these stages are also depending upon the person's age, whether it is a child or adult, sex, whether male or female, immunosuppression. What do you mean by the immunosuppression? So sometimes, for example, you want to kidney transplant, so immunosuppression drugs are given, so your immune system will be suppressed, and you can do the successful kidney transplantation, as well as the liver transplantation, as well as other organ transplantation. So in this case also the stages may be the vary, or their timing may be the vary. So co-infection with the other viruses, if already there is an infection with the other viruses, so these stages may be the vary. So pathology, here you can see that the all these antigen systems, so three antigen antibodies systems are there, and these three systems plays very important role. We can detect them, and if we found these antigens, and this antigen will be very good marker, or if we identify, then you can understand the phases, whether it is a window phases or what is the stage of the viruses. So these are the indicator whether they are replicating or not. For example, if E antigen is observed, then we can see that it is indicator of active replication of hepatitis B viruses. If okay, so it is a marker. So in this way we can also predict, and here you can see the response after the week after the exposure, how titer of antigen and antibodies, how it, interaction symptoms is takes place. I will not explain in the detail. Later on you can read in books. Pathogenesis is the in the pathogenesis what will happen. Now there will be the blood flow. Now this virus will enter the human body via body by skin mucosa via the blood flow enters the liver and other regions such as pancreas, bile duct, vessel, WBC, bone marrow, like that. And thus it will be all these antigens will be appear on the liver cells, and with infected with the uh, we can detect, and this will lead to the cytolysis of the liver cell. That means breakdown of liver cell. So in this way, cytopathy will be observed. That means cirrhosis of the liver cells will be breakdown, and there is a some characteristics of the liver will be changed, and that is called as a cytopathic effect. That cytopathic effect we can check under the microscope. We can change the physiology. We can change the size, shape of the viruses, and abnormality will be observed. Okay, and because of the viral infection, there will be the change in the structure of the host cell. Okay, so that kind of the cytopathy effect we can see under the microscope. What kind of the signs and symptoms we can observe? Fever, fatigue, loss of appetite, nausea, vomiting, abdominal pain, dark urine, joint pain, jaundice, and hepatomegaly. So in this way, these symptoms can be appear, and as it can begin an average of 3 months to 5 months after the exposure of the HBV. Okay, and these symptoms can be remain for several weeks, but it can persist up to the 6 weeks. So this is an incubation period, and this is the average days, and this is a range which is given. I will not explain it in detail. So these are the incubation period 45 to 120 days for the hepatitis B, and these are three important phases, that is pre-acteric, acteric, and convalescent. So in this way, these are the different symptoms appear, like nausea, vomiting in the pre-acteric, and in acteric also dark urine color is changed, and then based on that jaundice will be appear, that we can easily identify that phase. Then convalescent phase, we can different types of the antigen we can check. Then chronic hepatitis B also observed in 5 to 10% cases of the adult, but it is more common in the children, 70 to 90%. So in this way, this is a flow chart, and what will be the outcome of the acute hepatitis B. So finally, if it is well developed, then there is a chances of the disease that is called as a cirrhosis or hepatic cell carcinoma. Okay, so these are the acute hepatitis B, and that is not good for our health. So diagnosis can be done with the help of the markers, and these markers are nothing but the antigens. So we have to detect this antigen, that is surface antigen, core antigen, as well as the secretory antigen, that is a E antigen. So we after the detection of this antigen, if it is appeared, then we can easily understand there is a infection to the person. So in this way, we can find out the concentration of hepatitis B virus in the various body fluid. It is present high in blood, serum, and wound exudes. It is also moderately present in semen, vaginal fluid, saliva, and low or sometimes not detected in urine, feces, as well as sweat, tears, and breath. So in this way, we can another important thing is prevention. Prevention is very important. So primary prevention, that is we have to make the aware to people, and we have to give the vaccine to the people widely to prevent the vaccine. Then we have to implement the good blood safety strategy. Okay, we can also need to take the care of the health cares and community settings, and this virus how it should not be transmitted, we have to take the care. Okay, then the good practices like the injection practices, we have to follow good safe injection practices. Safer sex practices we have to follow. Okay, all the drug users, they have to also take care. All these things important. Safe food, water supply is again the important, as well as the education is also important for the healthcare workers. So vaccination, this is a vaccination history. We can give the vaccine to the people, but I will not explain in the detail, but I will tell about the vaccine, that is a plasma derived vaccine. So all of know that the plasma derived vaccine, in this way the person is a positive and now it is cured, we can take the plasma of that person, that will be the positive donor, which will consist, their plasma will consist the this viruses. We can inactivate these viruses if present, and then we have to formalin inactivated. That means we have to inactivate this virus either by using the formalin or the heat treatment, and then now that virus will be inactive. Okay, so virus live dead, it's not like that. Virus active or no inactive. So you have to make the inactive viruses from the plasma of the infected or positive donor. You have to take from their blood, and then you can inject into the another person as a vaccine. So a combination of the vaccines can be done. The vaccine that is a hepatitis antigen S vaccine can be combined with the Colamide Gorin bacillus, that is a BCG vaccines, measles, mumps, and rubella. And in this way, we can also protect our health by injecting them with the diphtheria, tetanus, and pertussis vaccine, as well as the polio vaccine. So in this way, the neonatal or newborn child, they can be protected, and we also immunize the mother. Secondary prevention, that is the effective diagnosis and medical support, that is important. So perfect diagnosis, and we have to keep free from alcohol, tobacco consumptions, and avoid the certain drugs that are toxic to the liver. In this way, the we can give the good treatment to the good support is necessary to the patients. Good nutrition is important, bed rest is important, and alcohol consumption should be totally stopped, because it is harmful to the liver, and it will help the virus again to grow properly and cause infection to the liver. So in this way, liver damage will be there if you are consuming the alcohol during that situation, and thus viral clearance is important. So avoid the alcohol. And then if you sometimes if this disease is progress, then there will be the fever, chills, headache, depression, as well as bone marrow suppressions, and rarely there will be the cardiac and the renal failure. Okay, so because of all that, so in the treatment you can give the different antiviral and immunomodulatory drugs. Okay, so these are the drugs which is listed that can be helpful, effective for the mm, to control the virus, and thus in this way we can um, uh, stop the infection of the virus, or you can prevent yourself. Tertiary prevention is that there is no surgical treatment for the hepatitis B. So you cannot remove or remove the part of the liver and all that. So no surgical yet treatment is developed. So in advanced liver damage because of the hepatitis and condition become life threatening, and there is a need a liver transplant. So what we can do, we can in tertiary liver transplant we can do it. Okay, so in this way we can protect our body. In the last conclusion, I will say that hepatitis B is the most common serious liver infection in the world. So it is caused by hepatitis B virus, and that attack liver cells and can lead to the liver failure, that is called as a cirrhosis carrying or cancer of the liver. And thus the virus is transmitted through contact with blood and body fluids that contain blood. So students, in this way, a liver is very important organ in the body, and if the liver is a fail or there is a problem, okay, so small virus can fail your liver and can develop cancer of liver, and then there is only one way, that is a transplant of the liver from the another person. So this was about the hepatitis A, hepatitis B, and HIV. So thank you, student, for your kind patience. Uh, Thank you very much.
Thank you very much, sir, for a nice presentation on viral infection.
you have covered this session in 2 sessions, and both the sessions were mostly knowledgeable. I think it is very difficult for the participant as well as the resource person, even though to sit in a post-lunch session. But we have enjoyed both these knowledgeful session. So will you remove this your PPT?
Yes, yes,
Stop
yes,
sharing PPT, yes, sir? Yeah.
yes.
Okay, okay. Thank you, sir. And please on your video. Yes. Thank you.
I think, sir, there is a little bit problem to my video, but I will try. But
Okay, sir. No issue. No issue, sir. No issue. No issue. Yes, yes.
I will try, but I think something there is a issue
Yes,
to
yes, yes.
camera.
Your camera, your system
Uh,
is not allowing your camera. Okay, sir. No issue.
Something setting has been changed. It is working, but
Okay, 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 sir.
okay,
I
sir.
think it is a hardware problem, I think, or the software problem somewhere, sir.
Okay, yes.
That's I think the topic is all clear for the participant, and if they have any query about this topic, because this topic, that viral infection, is not for the life science, but for our day-to-day life, it is a very important topic. And I think if the participant have any query or question over this, they can ask sir directly.
Yes, sir.
Sir, one HBV is curable or not?
Sorry, sir.
That hepatitis B, that infection is curable or not?
It is curable, sir.
Okay.
But
And
what happens, sir? But some person they have a habit to consume alcohol, to take drugs, and if their day-to-day life is not good, they are not taking good nutrition. Then what happens? So the acute hepatitis B will develop. We can control it, but once it is acute hepatitis is there, so there is a chances of cancer of
yes, sir, like
and cirrhosis, and in that stage then there is no any option, that means we have to change that organ, and then it is become again very risky to find donor and so many other process, and money is required. And that's why I always tell student that you are very rich because your brain is of 2 crores, your heart is of 5 crores, your
yes,
lungs are many. So you are very rich people, and we have to think positively, and we have to take care of this all this wealth. That's why health is wealth. Our primary goal should be we have to take care of our health.
right. Actually, what happened, sir? In one of my relative, uh, they had a surgery of kidney stone. Major surgery was there, and through that at that time they required a blood transfusion. Almost 3 bottles of the bloods were transfused to him, and later on, after 10 years, there was no history of another transfusion. But after 10 years, he has detected HBV, that hepatitis B virus. And now the doctor is saying that it cannot be curable. Either you go anywhere, but it is not possible to cure almost. But they are giving such a one kind of treatment line that the patient should be feel only well. But the patient, the doctor said that it cannot be cured. If you have any, yes, sir.
It is totally depending upon the person's age,
Yeah,
person's age,
yes, sir. The
and the
person,
medical condition.
yes.
So
One thing is there, sir. The patient age is only 21-year-old, and
okay.
the surgery is done at the age of around 12 years.
Okay.
Yes.
Then what was the cause for the infection? Was it because of the blood transfusion?
Yes, sir. It is due to the blood transfusion, because there is no history, genetic history of hepatitis B. And other infection, like either he didn't have any kind of jaundice in childhood or in other age up to 12 years. He didn't have a history of jaundice also.
So what happens, sir? It is because of the comorbidity. So comorbidity, that means kidney transplantation. Although kidney is a transfer, okay, so that is not 100% the part of our body. So we
Right.
adjust it, we compromise it because of our own benefits.
Yes,
What we did there, sir, we took the immunocompromised drug. And because of these immunocompromised drugs, what happen? You interfere with you are shut down your whole immunity.
Right.
And that is the reason during that time, what happened? This virus may have entered and it remained for a longer period of time as a integrated DNA, their DNA as a integrated in the host. And now what happened? Now that DNA is now everywhere.
Right.
Right now. And it
Yes.
is in window period as well as it is in window period as well as it is also showing some symptoms. So if some symptoms are there, you are going to the doctor. But doctor is saying that the already virus have trapped whole part of the liver, and that is
Yes.
he might suggest you have to another transplantation is important. But already the patients have transferred their kidney.
Yes.
So although it is a disease of liver, but it is very much connected with other organs also, sir.
Yes, sir.
Okay, kidney as well as our circulatory system as well as our immunity, everything it is interconnect with each other. If
Yes,
one
sir.
organ is affecting, so another organs are also affecting. They are interrelated.
Yes.
For example, if my abdominal ratio is changed,
Yes.
I have chances of type 2 diabetes. Later on, this type 2 diabetes may also converted into the BP. Later on, it affect my heart. Heart attack
Yes.
chances are also there. So,
Yes.
and kidney failure chances are also there because of
Yes.
diabetes. So, like that, there is so much interconnected, and that complication may be arised, and that's why doctor is saying that like that. That is the reason.
Right.
Okay, so it is not only a question of only hepatitis B. So, other organs might be also affected because of the
Yes.
infection.
Exactly, sir. Exactly.
That's why he's not saying that the hepatitis B cannot be eradicated, but maybe multiple organ disorder will be there. That's why he's
Yes.
saying it is not curable now because the
Yes.
organs are and other organs are also
Infected. Yes.
not working properly.
Yes.
Yeah, because kidneys are affected, no. So
Yes.
other organs are related to with that. That is the reason.
Yes, sir. Another thing, the fatty liver may convert it into the hepatitis disease.
So directly we cannot say that the fatty liver will be, but what happens, sir? We have to take care of whatever we are eating. Because everything is goes to the liver for the detoxification process. For example, if I am eating
Yes, sir.
pesticide containing fruit, so what will happen? So this pesticide may be carcinogenic to our body. For example, if I am drinking some cold drinks, so in that cold drink they are adding insecticide to control insect and small to our
Yes.
the growth of
Germs.
insect.
Yes.
So we are happily drinking these cold drinks, but we never think that is it good for my health. So automatically what happen? It is affecting some factors are also affecting to the liver. Okay. For example, if person's weight is from childhood is large, so the person's liver as well as heart is well suited to that body. So even person is a fatty person will not get heart attack. But even the person is slim trim, he will get the heart attack.
Yes. Right, sir.
Yes, sir.
Yeah.
So whatever the
Thank you.
genetically we got, it is a beneficial. But if we are doing some changes, our lifestyle and something, so we are inviting them.
Yes, sir. Exactly. Thank you very much, sir. You have a nice presentation on viral infection in this countrywide classroom teaching program, and it is our prime duty to say to thanks. So I invite my colleague, assistant professor in microbiology, Dr. Deepak Bhusari sir, to propose the vote of thanks for this session. So you have taken tirelessly continuously two sessions in this countrywide classroom teaching program, and you have covered a very important topic, which is important for everyone, not only the BSc student, but for all the general public. It is an important topic you have discussed in this countrywide classroom teaching program. And definitely, sir, this recorded video, which is available in our official YouTube channel, DBC Media Center, will be circulated among all the armed public. With this, I request Dr. Deepak Bhusare sir to propose vote of thanks. Dr.
Thank you,
Deepak
sir.
Bhusare sir, over to you.
Yes, thank you, sir. Before going to cast vote of thanks, Mr. Nikhil has raised his hand for asking something, I think. So, Mr. Nikhil, you want to say something or you have some doubt? So you can ask.
No, sir, no doubt.
Okay, thank you. Thank you, Chavan sir, for giving me this opportunity to cast the vote of thanks. Really, sir, Dr. Ashok Bankar sir has presented very nicely in his presentation, because in this present scenario, in this COVID-19 pandemic situation, that everyone is facing some major problem, because that no one aware about the what is going on here, because most of the people, they are not aware about the how viral infection is going on actually. And by this way, we can aware how this viral infection that occurs. And Dr. Ashok sir has presented very nicely in his presentation that I think the motto of this presentation, or this in this countrywide classroom teaching program, is that this viral infection, that is most important, that precautions is better than cure. That if we know about how this viral infection occurs, or what are the symptoms that occurs when this viral infection, after viral infection, so it may be is easily curable, because most of the people, they are unaware about that how problem. And in this most of the people, they are not getting what is actually happens with their self, because the peoples, they are facing that normal problem, but the most major problem is here, that is the breathing. That after some after viral infection, that Ashok sir has presented very nicely, that the structure of virus, that how that surface protein receptors that present. And as an antigen, when the viral that any foreign particle, they when entered in the human body, so after that, what are the possibilities and how they are infect to our cells? That is mode of action, their prophylaxis, that preventive measures. And that sir has very nicely presented all these the aspects regarding the viral infection. Sir, I'm very much thankful to on behalf of our total team of countrywide classroom teaching program, as well as I'm also thankful from our participants also for giving such a nice speech to all of us, because this will be a very useful for those who are the science students, because they the science, if the science students, they will listen, they will carefully listen or see this video or your presentation, then they will aware about their family, and in this way that spread the awareness about the viral infection. So here I'm once again thankful for giving your valuable time for giving such a nice presentation in this countrywide classroom teaching program. Thank you, sir, and hope for in the future you will also that you are giving the positive response to our all activities. Thank you, sir. Thank you once again, and thank you to all the participants who have joined with this live program for listening this presentation on viral infection. Thank you once again.
Thank you, Nikhil sir and Arvind sir and your whole team. Thank you once again.
Thank you very much, sir. And the session is concluded here. Tomorrow we will be tuned at morning 10 or 11 o'clock. Tomorrow we have 2 sessions. Thank you very much.